Okay, in today's lecture, we will be discussing the cell differentiation process. So, before we move on to the cell differentiation, let us recapitulate what I have shown in last one or two lectures. So, essentially cell differentiation is one of the cell fate processes, and other cell fate processes include cell replication, cell death, cell motility or cell migration and cell adhesion. So, and all these cell fate processes, they have direct relevance for the tissue function. So, if a tissue functions its normal functionality, then it is called homeostasis. Homeostasis means, homeo means same, and homeostasis means for the self survival of the tissue, the functionality of a given tissue is known as a homeostasis. Tissue repair in biological term is known as wound healing, and tissue formation is known as morphogenesis, and it is part of a development of biology. Now, as I have explained to you earlier that cell communication via different cell signaling processes are important in all these cell fate processes like replication, differentiation, death, migration and adhesion. Now, as differentiation is known as the differential gene expression or differential cell functionality, we should first know that what is gene and genome. So, therefore, this slide provides you some important or useful information. So, gene is actually nucleotide sequence in a DNA molecule. DNA stands for deoxyribonucleic acid. So, nucleotide sequence in a DNA molecule that functions to synthesize protein, structure RNA and catalytic RNA molecules. So, what is the function of DNA? DNA's function is to synthesize protein, and what is how this protein synthesized, that I will come to little later. That is via formation of RNA, and then from RNA the protein is synthesized. Now, structure RNA, catalytic RNA molecules, these are like different types of RNA molecules. And genome, what is genome? A complete set of information in an organism's DNA is known as genome. Okay. So, gene is the nucleotide sequence in DNA, and genome is the complete set of information that are contained in a gene in an organism's DNA. So, organisms differ from one another because their respective DNA molecules have different nucleotide sequence. So, two organisms, they are different because their respective DNA has two different nucleotide sequence. And what is this nucleotide sequence? Typically, this is that important slide. So, this is DNA structure. Now, if you look at from the top of the slides here, top left. So, what is the building blocks of DNA? Building blocks of DNA are phosphate, sugar phosphate, and there is one base. So, this is called base pairing. So, this for sugar phosphate plus base, that is what is known as the nucleotide. So, when I say the nucleotide sequence means that this phosphate sugar or sugar phosphate, that remains the same, only your base that is different. So, this base is here, it is mentioned as G. G stands for, so there are four types of bases: A, T, G, C. A for adenine, T for thymine, G for guanine and C for cytosine. So, these are like four bases. Now, A, T, G, C, and the way this base pairing takes place, it is like A pairs with T, that means adenine pairs with thymine, C pairs with G, that means cytosine pairs with guanine. That is a typical base pairing that is observed in a typical DNA sequence. Now, I repeat, now if you remember that what is the protein structure, what is the building blocks of a protein structure, that is amino acids, right. Now, two amino acids, they come together, they join via the peptide bonds, and then they form what is known as polypeptide, and these polypeptides, they can interact, they can fold and all those things, and then they can give different shapes of a protein structure, be it primary structure, be it secondary structure, be it tertiary structure, be it quaternary structure. So, similarly, a DNA molecules, the base structure or base is known as the nucleotide. Base structure means that is that building blocks of DNA is known as the nucleotide. Now, what is this nucleotide? Nucleotide contains one phosphate sugar molecule. So, phosphate molecule means PO4 3 minus, and there is a sugar molecule, and this phosphate sugar, that forms the backbone kind of a structure, just like any polymeric molecule, CH bonds, they form a backbone kind of structure. Similarly, for DNA, it is the sugar and phosphate that forms the backbone structure. and in this backbone structure you have 4 different bases. I repeat, adenine, thymine, guanine and cytosine. Now, these are known as ATGC, and so this is your one backbone here, this is your another backbone here, in between this is known as the base pairing. Okay. Now, this base pairing, as you notice that there is very specific base pairing. A always pairs with T and G always pairs with C. That means A would never pairs with G or A would never pairs with C. Okay. So, AT is possible, GC is possible. Okay. What is not possible is that AG or AC. So, and another thing is that so these are 4 different. Now, why 4 different nucleotides? The moment you replace G with T or A or C, then this nucleotide also would be different. Okay. So, similarly, in a protein molecule, when you have these polypeptide chains, now the moment you change one R radical in one of the amino acids, then entire protein molecule composition and configuration also will change. Similarly, here you have this one is the backbone and one is the base pairing, one is the G here. So, the moment G is replaced by C, that is a complete different nucleotide. Okay. The second thing is that now this is known as the individual strand. Okay. So, this is one strand and another one is that this is second strand. So, this is strand number 1 S1 and this is strand number 2 S2. So, this backbone in biological terminology it is known as the strand. So, that is why it has been written that DNA composed of 2 DNA strands, long polypeptide chains, and these 2 strands, in between these 2 strands you have the base pairing AT and GC, AT and GC. The third one is more important. So, now you know that DNA composed of 4 nucleotides, it has a strand, this strand is made of the sugar and phosphate molecule, and there is base pairing, there is specific combination of base pairing AT and GC. Okay, that is good enough. Now, second thing that is important here is that this complementary base pairing enables packing in energetically most favorable arrangements. Energetically most favorable arrangement means for any stable structure, it has to minimize the total energy of the system, that is the law of nature, right. So, here these DNA molecules, if they have this double helix kind of a structure, then that is been biologically shown as the energetically most favorable structure. So, DNA molecule stable structure is not this one, but DNA molecule stable structure is this one. Now, how this structure? They just wind around against each other, this DNA structure, and what you see here that to maximize the efficiency of base pair packing, 2 sugar phosphate backbones means 2 strands wind around each other to form a double helix with one complementary turn, with one complete turn every 10 base pairs. With one complete turn means, suppose from here to here, how many are there? 1, 2, 3, 4, 5. Then similarly, in next one is that, so the one turn means the moment it comes and makes the complete turn. Okay. So, this complete turn, in this complete turn you have 5 here, again there will be 5 down. So, 10 or 5 up would be there. So, that would be 10. So, total 10 base pairing makes one DNA strand make a complete turn. Okay. So, that is the way these structures are determined. Is it clear? So, here what is that? Now, again all this nucleotide sequence, it would be difficult for you to remember, but to show you that what is the typical organic structure of this ATGC, that is the base pairs here. So, T is here thymine. What you see in this base pairing is that in case of the T, you have a typical benzene like ring here, and but here it is not all carbon, some carbons are replaced by nitrogen, and there also there is some double bonds, and then some hydrogen and oxygen and hydrogens are attached to each of the carbon atoms. Okay. Now, another thing that you notice here, in this base pairing between A and T, there is hydrogen bonds are there. So, this is like non covalent bonds or weak bonds. Between C and G, that is also again hydrogen bonds. So, therefore, what you notice here, that this AT and CG, this base pairing by hydrogen bonds, and hydrogen bonds, one of the uniqueness of the hydrogen bonds is that it can be broken if it is so desired. Okay. So, shapes and chemical structure of bases allow hydrogen bonds to form efficiently between A and T and between G and C. That is what is known as, that is what is the uniqueness of the structure. So, AT and CG, this is known as the complementary base pairing in DNA double helix. The other things in the DNA structure that I have mentioned is that uniqueness. Each strand in a DNA structure acts as template or mode for synthesis of a new complementary strand. For example, if we designate S and S prime as two strands, then S can serve as a template to make a new strand S prime, and while S prime as a template for making a new strand S. Now, let us go back and try to understand what it means. Suppose this is strand S, this is strand S prime. So, what it means is that the strand S has a unique property, then it can make a complementary strand S prime. Similarly, S prime individually has the capability to produce a complementary strand of S. So, this individual strand S and individual strand S prime can serve as a template to produce the opposite strand either S prime or S. Is it clear? So, this S and S prime can serve as a template. So, that is the uniqueness of the structure, and then ability of each strand of a DNA molecule to act as a template for producing a complementary strand enables a cell to copy or replicate its genes before passing them on to its descendant. So, what it means is that, now look at this slide and try to understand. Okay, first of all, as I said that between A and T and C and G there is hydrogen bonds. Okay. Now, if required, these here laterally these hydrogen bonds can be broken. Now, to S, what are the bases that will be attached? Two nucleotides, one is T and one is C. To S prime, what are the bases that will be attached? One is A and one is G, right? You understand? So, now S has the capability to produce a complementary strand as S prime, and S prime can produce another complementary strand as S. Now, only thing that they require is that opposite bases. Now, T is there, C is there. The moment they find A and moment they find G, so simply A and G can be fixed to the S prime and can form another DNA molecule. So, that is what has been mentioned here, is that the ability of each strand means that is either S or S prime of a DNA molecule to act as a template actually enables for producing a complementary strand, enables a cell to copy or replicate its genes. So, gene replication or copying is possible because simply because S can produce S prime, S prime can produce S, and secondly because between A and T and G and C there are weak hydrogen bonds which can be broken if it is desired. Okay. For example, DNA replication in a bacterial genome. Now, bacteria, you know that DNA, RNA, all these genetic materials, they are not contained within a nucleus, but they are just dispersed in a cell. Now, this bacterial case, for example, this is a replication origin here. Now, this replication origin here you can see that it will start, this green one, it will start to grow, and subsequently these two daughter DNA molecules will be formed. And for bacterial cells, typically it takes around 40 minutes. So, this entire length time span is that T is equal to 40 minutes or less than an hour for two daughter DNA to be produced. And that is the reason typically bacterial cells, for the proliferation and growth, we check in the laboratory for 4 hours. 4 hours means you give approximately 8 times than one individual 2 daughter DNA molecules to produce. And then this 40 minutes, as you can see here, it duplicates its genome of 4.6 into 10 to the power 6 nucleotide pairs. So, in these 40 minutes, huge number of nucleotides they normally are produced, and this is of the order of 10 to the power 6. In the beginning, what we have mentioned is that DNA molecule, that what is the role of the DNA molecule? It produces or synthesizes a protein, structure RNA and catalytic RNA molecules. And then other things is that genes carry biological information that must be copied accurately for transmission to the next generation each time a cell divides to 2 daughter cells. What it means? From one cell, if it goes to 2 daughter cells, that this each daughter cell must have a copy of its parent gene very accurately or precisely. The second thing is that a typical human cell contains 2 meter long DNA. So, 2 meter long DNA means it is more than a person's height, right. So, if a guy is 6 foot tall, then it is how much? It is around 180 centimeter or around close to 2 meter. So, more than 6 foot height, a person's length is there in the DNA in the human cell, a typically human cell. But how this is possible that 2 meter long DNA can be squeezed? Because it has a double helix kind of structure. So, that is what is required to make this energetically most favorable structure. You can squeeze it, but you can if you stretch it and put it all together, then you will find that it is a kind of 2 meter long DNA structure. And how many nucleotides and how many different proteins it has? It has almost like 30,000 different proteins that it carries the instruction. Now, let us see that how this DNA can form the proteins. You have this S and S prime stand, and here you have this AT or GC kind of base pairing. Okay, this is it, and this red one is all the hydrogen bonding here. Now, what happens during that from DNA to RNA? RNA is ribonucleic acid and DNA is deoxyribonucleic acid. Now, what happens from DNA to RNA? Just one strand, and this, what is this strand? This 5 prime and 3 prime, that strand is getting detached from the DNA molecule. And if you see closely that whatever 5 prime 3 prime, that whatever this bases were attached to this, and same bases were attached here also. Okay? So, it is just that this entire DNA molecule has been transformed to one RNA molecule. So, this type of DNA to RNA, this is known as transcription. Transcription means, so this transcription means the nucleotide sequence in a chromosome is first copied into RNA, and this process is known as the RNA synthesis or transcription. Now, here the proteins are not formed. Proteins are formed in the next stage when RNA goes to the another process called translation. So, translation is the stage where the proteins are synthesized, and you can see here that this is the long polypeptide chain, this is C terminal and this is N terminal. Okay. So, that means this long polypeptide chain molecule has formed, and this is that exactly the flow of. So, you have to take a note of it, that irrespective of the cell type, in all living cells, this is the way proteins are synthesized. DNA goes through transcription to RNA, RNA goes through transcription to translation to protein. So, this is that and this is known as, in molecular biology language, it is known as the central dogma. Central dogma, what is central dogma? Central dogma in molecular biology language is known as the process of DNA transcription to RNA and from RNA to protein. Okay. Now, the question is that how this thing happens, this now what are the different parts of the RNA? what are the different types of RNA? One RNA you must be knowing is that mRNA. What is mRNA? That is messenger RNA. Okay. Now, DNA you know, so suppose this is an eukaryotic cell, okay, and you have a nucleus here, you know this nucleus has a nuclear pore complex. Now, DNA you have in the nucleus. Now, DNA to RNA synthesis therefore must happen inside the nucleus. Now, this RNA once it is synthesized, that is once it is transcripted, it goes through certain processing stages which is known as RNA splicing. RNA splicing means the simple RNA, then it goes to mRNA, but the way I am writing here, it is not that simple. The biologically the RNA being processed to a more refined step called mRNA before this mRNA goes outside the nucleus to the cytoplasm. You understand the whole process? So, DNA, so up to this process DNA to RNA, this is all takes place in the nucleus. Up to this process RNA to protein synthesis, this all takes place in the cytoplasm. Now, how this thing happens? DNA to RNA transcription happens in the nucleus, then RNA goes through RNA splicing or some processing stages to transform to mRNA. Now, this mRNA now goes to the cytoplasm. This mRNA now goes to the translation stage, okay, and then forms a protein on the cytoplasm or on the cytoplasm matrix. Remember, in the cytoplasm you also have certain organelles which is known as the ribosomes. Ribosomes is another way, another place where proteins are synthesized in large number. Okay. So, now let me summarize what I have said on this slide. This slide is important. So, what is the role of DNA? DNA actually contains genetic information, and DNA's role is also to synthesize proteins and RNA. Okay. So, DNA has a double strand S and S prime, and DNA has 4 different nucleotide sequence A, T, G, C. Okay. Now, in this base pairing, when DNA transforms to RNA, what happens? One of the strand, either S or S prime, along with the different nucleotides, they get detached from the double strand and then they form the RNA molecule. Now, this RNA molecule DNA to RNA synthesis is known as the transcription process. In the subsequent stages which take place in the cytoplasm outside the nucleus, these RNA molecules, they undergo translation process and then they make the proteins, and this is the typical protein structure. Right hand side, as far as that convention, it is C terminal, left hand side it is, as far as the convention, it is N terminal. Next question, that what is the biological difference between DNA molecule and RNA molecule? Okay. Now, let me try to answer this one. Biological difference is that DNA has a double helix kind of a structure. Okay. Now, RNA has a single strand. So, RNA does not have a double strand or double helix kind of a structure. Now, DNA, it has a typical base pairing of AT and CG. RNA has also 4 nucleotides, but typical base pairing would be AU and CG. So, in case of RNA, T thymine is replaced by U. What is U? U is known as uracil. That is another different base. U means uracil. So, in RNA the base pairing would be AU and CG. So, this is the second difference. Yeah, third difference is that in nucleotides in RNA are ribonucleotides, they contain sugar ribose rather than deoxyribose. That comes from the basic nomenclature itself. So, since DNA is the deoxyribose, so they are the nucleotides are the, they contain the deoxyribose. In case of the RNA ribonucleic acids, so they contain the ribose, that is the sugar molecule. So, this is the major difference between DNA and RNA. Now, coming to the genes with different deficiencies, let me explain that that although the transcription and translation process takes place on different genes, but two different genes, let us say A and B, they can have different efficiencies. Efficiencies means one gene can produce large number of protein molecules, whereas other genes can produce very small number of protein molecules. Now, how it happens? Now, in this particular case, gene A, when it goes to the transcription, the RNA molecules, if you see, a number of RNA copies are made. Now, these individual RNA copies, then they can individually produce a large number of protein molecules, and as a result you end up of having capital N number of protein molecules here. Now, in case of gene B, although it produces the same similar RNA copies, but the number of RNA copies is just 1, and therefore, you have very small n number of protein molecules. So, therefore, gene A is known as highly efficient, gene B is known as less efficient. The second thing is that in biological language, if you consider that the gene A can be expressed, or expression of gene A would be much more than expression of gene B. What it means by expression? Expression means here how much protein molecules a particular gene can produce via transcription translation process. Okay? So, therefore, this entire biology of this transcription translation leads to more gene protein molecules formation in gene A. So, their gene A has the higher expression. So, here it is mentioned that gene A is transcribed and translated much more efficiently than gene B, and this allows amount of protein A in the cell to be much more greater than that of the protein B. So, essentially what it means here, gene A ultimately will synthesize protein of type A, gene B would be ultimately producing the protein of type B. However, quantity wise A protein is much larger in numbers than B protein. So, although the message that I am trying to send, although gene A and gene B, they are contained within the same DNA, but individually gene A and gene B has different efficiency because of their any difference in the base pairing or some other biological properties. Now, from that you can say that that now I can start explaining what is cell differentiation. So, differentiation means, as I said earlier, that is the differential gene expression. Now, differential gene expression means that how two different genes they are expressed differently, you have the idea, like through transcription and translation process, and because of two different efficiencies. Now, therefore, suppose this is cell A and this is cell B. If I say cell A has different gene expression than cell B, that means that in the A cell, in the nucleus DNA, if both are eukaryotic cell, in the nucleus DNA they have different efficiency of protein synthesis compared to gene in cell B. Okay? So, ultimately the function of these genes are related to the function of the protein synthesis. So, their first thing is the definition. What is definition? Differentiation is a process by which a cell undergoes phenotypic changes to a specialist cell type in terms of physiological function. So, therefore, two cells, if they are differentiated, that means A has a different physiological function than B, and why they can perform two different physiological function? Simply because their DNA structure, their gene structures are different, and their efficiencies, the efficiency of both the genes are also different. The process begins with a lineage commitment. What is lineage commitment? I will come back to later. Followed by a coordinated series of gene expression events before being differentiated for specific function. Now, let us see that what is meant by lineage commitment. So, this is, you start with a stem cells here, mesenchymal stem cells. Now, what is mesenchymal, what is stem cells, I will take a different, I will explain maybe later if time permits. Now, the stem cell is the primary cells. You have to remember only one thing at this point is the stem cells is the primary cells. All the other cells, they are known as the transformed cell. Transformed cell means that cell must have transformed from some primary cells via differentiation process. Now, if you see that stem cells here, the primary cells, it goes to proliferation. Now, proliferation, you can have osteogenesis, you can have a chondrogenesis. Osteogenesis means it is forming the bone cells. Chondrogenesis means it forms the chondrocytes or cartilage tissue cells. Now, commitment means, if you remember that cell cycle, I have shown you that when a cell goes from S1 to G and G to S2, there is a checkpoint. Checkpoint means that the cells are now ready to enter from S1 to G. So, now once a cell is committed to enter to G, it will remain in G, it cannot come back to S1. That is the simple logic. Okay. Now, similarly, here the commitment means once the cell is committed to differentiation, that means it will go to first transitory osteoblast, then it will be differentiated to osteoblast. Now, transitory osteoblast and osteoblast cells, they have different gene expression or they have different cellular functionality, and then it will go to more matured cells, is that called osteocytes. It is like you can always consider or you can always compare this with the development of a child. As the age goes up, a child becomes more and more matured, and that you can see in their expression. They can learn the things, they can understand things, they can respond to different things, right. Similarly, here the moment transitory osteoblast forms the osteoblast, they can form bone cells, but it is more matured stage is the osteocytes. So, some of the physiological function that osteocytes can perform, osteoblasts may not be able to perform, because osteocytes is a differentiated form of osteoblast. I repeat, osteoblast and osteocytes both are bone cells. However, osteocytes are more differentiated stage of the osteoblast, therefore, they are different gene function. Similarly, here in case of chondrogenesis, you have transitory chondroblast and then you have chondrocytes, and then you have hypertrophic chondrocytes. Hypertrophic means atrophy, hypertrophy. Hypertrophy means increase in the cell size. So, similarly here, so when you say the hypertrophic chondrocytes means simply chondrocyte cells are increased in size. Okay. Then similarly, there is a myogenesis, there is a myoblast cells, then myoblast fusion. Myoblast fusion means two myoblast cells coming together, and then you have the myotube. So, similarly, you have this dendrogenesis or ligamentogenesis. Ligamentogenesis means it is a formation of ligament. Dendrogenesis means it is a formation of tendon. Now, you have the fibroblast cells here. Now, this fibroblast, it is the cells of the connective tissue. Now, all these fibroblast cells together with the ligament, and then it forms a ligament cells. So, this is known as the lineage commitment. Lineage commitment means that a cell comes from its origin. It is just because if you look at the family history of a person, any person is coming from his ancestor, right. So, and then it comes like grandfather, great grandfather. So, similarly here in the cell case also, osteocytes, father is osteoblast, grandfather is transitory osteoblast, and it all comes from the stem cells. So, stem cell is the primary cell where it can be differentiated into many cells, and all these cells, once it is differentiated, their genes are also copied, and they have a different gene expression. So, other things in the differentiation process is that suppose at any stage osteoblast cells, okay, is divided into two daughter osteoblast cells, then their gene expression would be because their two genes are copied here from one single DNA. If osteocyte cells, if they are divided into two osteocyte daughter osteocyte cells, again they will have same gene expression. Okay. So, differentiation, division, proliferation, adhesions, they are conceptually different meaning. That is what I am trying to repeat here. Okay, that occasionally mature cells can undergo changes to another cell type through transdifferentiation by specific molecular activation. Now, I will possibly give an example on that. Then fourth one, this complex differentiation process can be defined in terms of gene expression and cell function. That is what has been already realized by you. So, different gene expression and different cell function, that is what is known as the cell differentiation. Differentiation involves the expression of unique genes that are specific according to a cell type with an irreversible change towards a particular cellular phenotype process. What I mean is that osteoblast can be differentiated to osteocytes, and it is a irreversible process. So, if you go back to that, so this thing cannot take place, osteocytes to osteoblast. You understand what I am saying? So, what I am trying to say that osteoblasts can only be differentiated to osteocytes in a irreversible manner. It is not a reversible phenomenon, this differentiation process. This is like very kind of bookish type of definition, that is involves a expression of unique genes specific to the specific cell type. That means, for osteoblast it is a different genes, for chondrocytes it is a different genes, for fibroblasts it is a different genes which are responsible for the differentiation process. Differentiation also involves a carefully orchestrated switching off and on of gene families. What it means is that many genes are inactive, but the moment it gets certain signal for cell differentiation to take place, those inactive genes are switched on. So, they become active. Now, once the differentiation process is over, then again they become inactive. So, it goes through switching off and switching on type of process. The final set of genes expressed are those that pertain to the function of the differentiated matured cell, and for eukaryotic cell differentiation is irreversible in nature, that I have already mentioned to you. So, always you remember the differentiation is irreversible in nature. Okay, we are discussing the differentiation of the cells. Some of the key points to remember is that once there is a commitment for a given cell to differentiate to different cell types, then it follows a lineage. That means that osteoblast cells will be differentiated to osteocytes in a very irreversible manner. So, this goes from top to down, not from down to top, that is not possible. So, similarly chondrocytes, that is the cartilage tissue cells, they will go to the hypertrophic chondrocyte cells, myoblast cells will go to myotube and so on and so forth. There is another type of differentiation that is called, there is two type of differentiation, one is called normal differentiation, another one is blocked differentiation. So, block differentiation is what happens in case of the in vitro study. In vitro means that is the culture medium in a laboratory. So, here differentiation is limited by the needs to proliferate. So, there are two things here, one is the differentiation and one is proliferation. This differentiation is limited by the needs to proliferate, and the population becomes predominantly progenitor cells, although stem cells may also be present. Now, what is shown in the right hand side, this bottom here. So, there are two cells which goes from one cell to one type to another type. Now, in 18 to 72 hours, then these two cells will start getting differentiated to different types of cells. As you can see that 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16 and so on. And then subsequently that after this amplification, so this is attenuation and this is amplification, that one particular cell type goes to another particular cell type by a differentiation process. So, this is one type of differentiation, what is known as a block differentiation. In the normal differentiation case, it's small stem cells, as I said, the stem cells is the primary cells. Now, this a small stem cell gives rise to a proliferating compartment that produces the differentiated cell pool. So, this is the case for the non-proliferating differentiating cells. So, non-proliferating means they are not proliferating, but they are just differentiating. And here this one is that called terminally differentiated cells, where it is almost at the terminal stage and it cannot be differentiated any further. In the second stage here, you see that if I call it stage 2 and this is called it stage 1, this is called committed progenitor cells. Committed cells means these cells will now be bound to undergo differentiation process. It's just like, as I said, in the cell cycle, it was going from S1, G, S2, M. Once it comes from the checkpoint between S1 and G, the cell is committed to enter into G, and once it is goes to checkpoint from G to S2, cell is committed to enter into S2 phase. Similarly, this commitment means now this progenitor cell must differentiate, it cannot go back to the earlier position. Okay. So, this is like takes place in a irreversible manner. This is that combinations of few regulatory gene proteins, as you can see here, that they can actually induce differentiation and form a large number of cells. So, this is the embryonic cells, it can be stem cells. Now, induction of the gene regulatory protein here 1. So, here this protein is already inside the cell, this gene regulatory protein. So, this cells is divided into 2 cells, daughter cells. Now, this is that induction of gene regulatory protein 2 and 3. Now, this 1 goes already inside, 2 is also coming here. So, 3 is coming here and 2 is coming here. Now, here these gene regulatory proteins are also coming in individual 2 daughter cells, cell C and cell D. Then again they will induce that other gene regulatory protein is that 4 and 5. Then similarly, these gene regulatory proteins also will enter each of the daughter cell. So, each time the cell divides into 2 daughter cells, the gene regulatory proteins are internalized in the cytoplasm of the cells, and this internalization process actually helps cells to further differentiate to many different other cell types. Now, this is an unique example what is known as transdifferentiation. I have mentioned to earlier that in the initial discussion of the differentiation. Now, what is transdifferentiation? Suppose you have that one type of cells, fibroblast, which is a connective tissue cells. Now, fibroblast cells can be transdifferentiated to smooth muscle cells. Now, what is the performance of the muscle cells? This actually helps in the contractions. Muscles means that can contract and expand, right. So, this kind of contraction, fibroblast cells can be transdifferentiated into cartilage cell, chondrocyte cell. Fibroblast cells can be transformed to bone cell, osteoblast and osteocyte. Osteocyte is the more matured differentiated cells formed from the osteoblast cells. So, osteoblast and osteocytes are both the same cell type. The osteocyte is the most matured and differentiated cell type. So, what you can see here that this is an unique example where fibroblast cells can be differentiated into 2 or 3 different type of cells. In one case it forms a cartilage cell, chondrocyte cells, another case it forms a smooth muscle cells, another case it forms a bone cell, that is the osteoblast cell. So, this is called the transdifferentiation case. Now, real time gene profiling. So, now as I said that that there can be So, as I said earlier that although the gene A and gene B, they are contained within the same DNA, but gene A and gene B can have different efficiency. Efficiency is measured here in the context of the gene is that in terms of how much or how many protein molecules that it can synthesize finally. So, here you can see from gene A, that is the RNAs are formed by the transcription. From translation from RNA to protein molecules, it forms a huge number of A protein molecules, but gene B only few B protein molecules are formed. So, what it means? That means gene A is highly efficient compared to gene B, or in other words gene A can be expressed more than gene B. So, expression in the biological terminology means that a particular molecule with how much concentration they can be measured. Suppose if I say that gene A has higher expression, that means that gene A can be measured with much more higher concentration by a certain analytical equipment. So, real time gene profiling means that when that actual differentiation process occurs. So, this differentiation process means that your gene expression changes. That how this gene expression can be changed? That changes can be monitored by two techniques. One is the microarrays and another one is the real time PCR. PCR stands for polymerase chain reaction. Okay. So, PCR stands for polymerase chain reaction, and these two techniques. Now, what is the principle of operation? I may be able to explain it this PCR, but not the other one. So, that what is that principle of operation of PCR? Then when I will explain, then you will be able to realize that PCR can actually be very useful to quantify that which are the genes which are expressed during the cell differentiation process. So, human genomes typically contain tens of thousands, typically estimates is 20 to 25,000 different genes, and every cell in human uses a subset of these genes at any point in time. That means that you take any cell what is present in living conditions, they just uses that out of 20-25,000 a few genes for its survival or for its functioning. Now, what is the gene expression changes? Changes in gene expression actually confer changes in the biochemical function. Biochemical function means that a cell it's always supposed to do certain function, like connective tissues or endothelial cells, they are supposed to provide support. Connective tissue cells, they can also form, they can support mechanically to the ligament and tendon. So, all these changes, all these functions are possible because they have a particular gene expression, and also many times changes in gene expression means changes in its ultrastructural features. Ultrastructural features means the way the cellular organelles are organized inside the cell. Some of the early changes in the gene expression involve changes in the transcription factors. What is transcription? Transcription is the DNA to RNA synthesis, okay? And regulatory proteins that are expressed. What is regulatory proteins? That can be metabolic regulatory proteins, that can be gene regulatory proteins, different type of proteins are expressed. Terminal differentiation means, as I said earlier, once the cells reaches terminal, anything terminal like a human being, it goes to a terminal stage means it cannot come back, right. So, it is almost towards the end of life. So, similarly here terminal differentiation means genes more specific to the mature cell type. That means, once the cells actually achieve most mature stage, cells cannot be differentiated any further. So, if you see that osteoblast lineage, it is the osteocyte cells which are the most mature cells. Osteocyte cannot be differentiated to any other bone cell type, that is the last stage of the differentiation. So, therefore, that is what is mentioned here, that during terminal differentiation genes more specific to the mature cell type are expressed. Fourth point that mentioned is that those specific proteins that function together to produce the cellular processes particular to that cell type and determines its phenotype. So, that means that from DNA to RNA to proteins by translation process, whatever say particular set of proteins that are generated, these proteins they function together to produce the cellular processes. That means, whatever the cellular functions that they are supposed to carry out. The next topic is the cell death process. So, in the cell death or apoptosis process, I will first give an overview of the cell death processes, followed by the biochemical mechanisms of apoptosis. Third one is that how to quantify cellular apoptosis process. Fourth one is that example of the bone cell apoptosis. Now, cell death is that continuous signaling by growth factors, hormones. So, these are like different signaling process, growth factors, hormones and cytokines and cell-cell contact and cell matrix interactions are necessary for cells to refrain from undergoing apoptosis, keeping them alive. So, what it means is that as long as the cells get or receive all the signals necessary for its survival, cells will survive. The moment the cells are deprived of these signals, cells will undergo apoptosis or death. It is as simple as that. If a human being is deprived of taking food every day, that automatically that human being cannot survive. So, similarly here in this particular context, the cells, if they are not getting survival factors or survival signals, then it will undergo apoptosis. Then what is necrosis? Necrosis is essentially mediated by the tissue damage mediated by hypoxia. What is hypoxia? I have told you earlier, hypoxia is that lack of oxygen supply to a particular cells, or that particular tissue has the inability to use the supplied oxygen for its own survival. So, either of these cases, this hypoxia actually leads to necrosis stage. Now, defects in the transmembrane transport lead to cell swelling and lysis. So, that is also another case of the cell death. During tissue development and function, programmed cell death or apoptosis that take place. Now, this apoptosis is the quiet mode of cell death. Many times you might have seen or you might have heard from people that a particular person undergoes silent heart attack. Silent heart attack means that there is not much enough symptoms earlier that the person will get a heart attack, and suddenly he sleeps and the next morning you will find that person is dead. That means the person had a very silent heart attack. So, you can consider that apoptosis is also very quiet mode or very silent mode of the cell death, and this apoptosis takes place in a very programmed manner. It is not a very irrational or illogical manner this apoptosis take place. So, many times if a cell does not undergo apoptosis, then what will happen? These cells, a number of cells, they can cause a tumor locally at that particular infected place. Now, apoptosis, like many of the scientific words, it is also origin of a Greek word. Now, this Greek word, the apoptosis means dropping off. Dropping off means in a particular season you might have seen that all the trees, their leaves are dropped off, right. So, these leaves are dropping off from the trees. So, similarly apoptosis means in the context of cell biology means the cells are also kind of dropped off, like they are not anymore dead. So, they are not anymore alive, or they are now dead. Now, what is that kind of textbook type of definition of apoptosis? It is a programmed physiological mode of cell death that plays role in the tissue homeostasis. Tissue homeostasis means that a particular tissue, as long as it is able to perform its own function, that is what it is desired for, that is called the tissue homeostasis. Alternatively, apoptosis is also described as the genetically encoded cell death program, which is morphologically and biochemically distinct from necrosis or accidental cell death. In the case of necrosis, what happens? The cell membrane is ruptured instantaneously, and then the cell undergoes necrosis process. This is more like a accidental death process. So, again coming back to the thing that I have mentioned. So, necrosis can be death due to road accident. Apoptosis can be described as death due to the silent heart attack. Okay. So, silent heart attack means it is a very quiet mode of cell death, that is the apoptosis, and necrosis means like road accident, that it is immediately a person died, and that is what is meant by necrosis process. So, this is that general language. It is, if we mention, then it is for you to very easy to remember. Now, if you look at this slide, and you would be able to realize that what are the morphological changes that takes place during cell lysis process. So, left one is that necrosis. As you can see that cell membranes are ruptured here at different places, and all the intracellular organelles, they are like coming out of the cell. Now, apoptosis, actually you do not see much changes in the cell membrane structure. So, that is cell membrane is more or less intact here, but cells, they shrink in size. Okay. So, cells shrink in size means shrinkage of the cells in the adaptation process. The biological term is atrophy, right. Atrophy is the biological term. Then there is membrane blebbing, that is also occurred, and third one is that many times the nuclear condensation or fragmentation means that whatever nucleus was there in a stable vital cell or live cells, the nucleus there becomes condensed, or many times they fragment, or there is fragmentation of the nuclear membrane. So, these are the kind of signs, morphological signs of the apoptosis, but in the necrosis one can immediately understand that cell membranes itself, they are ruptured, and all the intracellular organelles they come out, and then they form the necrotic cells. Sometimes in our lab we have seen that once you do the electric field experiments, if you apply too much electric field or electric voltage, the cells undergo necrosis, not the apoptosis process, because we can see that cell membranes are ruptured and all the intracellular elements are coming out. Now, there are 3 phases of the apoptosis. One is the induction phase, one is the effector phase, and third one is the degradation phase. Induction phase is that this is activated by certain death inducing signaling molecules. So, as I said earlier that for every cell fate processes you require some sort of intracellular signaling process. So, some death inducing signaling molecules are there. We are not getting into details of these molecules, but we are discussing the mechanisms here. So, in the first stage is this first stage is activated when the cells receive some death inducing signals. Second one is that effector phase. Effector phase means that this is the phase when cell commits suicide. So, as I said that it is also a kind of more like a suicidal mode, but in a very silent manner. Third one is the degradation phase. During the end stage of apoptosis, a cascade of caspases. Caspases is a particular signaling pathway that are activated, and loss of mitochondrial membrane function occur. Now, why mitochondria is important? Mitochondria is a place where ADP to ATP, ATP to ADP, all this transformation takes place, and mitochondria is known as the powerhouse of the cell, like it produces energy. Now, at any certain instance, if the mitochondrial membrane does not function, then what will happen? This mitochondria will not be able to produce the energy required for that particular cell, and therefore, it can lead to the cell death process. Clear? So, that is what has been mentioned here. This mitochondrial membrane function stops, or there is a loss in mitochondrial membrane function that involves a large opening in the mitochondrial membrane that may mark the point of no return in the process. The point of no return means now cells will definitely undergo death. It cannot come back anymore. So, it is from that point onwards, it is more like a irreversible process of going to death. So, the other things that I have mentioned now here, it is more clearly written that an external murder signal, that is the Fas ligand binding to the Fas surface death receptor, that induces apoptosis, and a suicide program that is initiated by an external stress such as the ultraviolet radiation. For example, if you expose some cells to a very ultraviolet radiation, UV radiation, the cells can die. That means, the cell apoptosis here is instigated or is triggered by the ultraviolet radiation. Removal of extracellular survival signal. So, either you induce the death signal or you simply remove whatever signals that were necessary for the survival of that particular cell. So, then also you can induce the apoptosis. So, all 3 pathways typically converge to a common family of enzymes that is called caspases, as I mentioned earlier. Once activated, they attack many proteins and ultimately resulting in the characteristic laddering of the DNA leading to the apoptosis. Little bit of mathematics here, but very simple. The apoptosis process resembles that of commitment to divide. If you remember that similar expression we have also seen in case of the cell division process. So, the rate of apoptosis, that means the number of cells that is undergoing death, dx by dt, is proportional to the population at any particular instance x, and the proportionality constant here is known as k. So, dx by dt is proportional to x, that means dx by dt is equal to x, and therefore, at any given instance x t you can written as that x naught exponential k t, and what is k? K is the rate of apoptosis. that is process proceeds by a first order kinetic process. So, this apoptosis is a first order kinetic process, that means that dx by dt is proportional to the x to the power 1. Okay, coming to the some examples of that cellular apoptosis, that how it happens. I will give an example that when a cell is treated with a particular particle eluates like hydroxyapatite mullite, how this apoptosis take place. So, this is that hydroxyapatite particles here. So, these particles are typically in the nano size range 100 nanometer or less, and there are different other particles that we have taken is a hydroxyapatite 20% mullite. So, there also the particle size is very small. Hydroxyapatite, that what we have used in our experiments, it has a Ca by P ratio of 1.67, that means it is more or less stoichiometric hydroxyapatite. Now, with some experiments, particular experiment I will show you that these particles, when these particles they are in the culture medium where you are culturing the fibroblast cells or osteoblast cells, how the DNA is damaged. So, this is a typical double helix pattern of the DNA. Okay. Now, when it is treated with biomaterial eluates, what happens, that DNA in the double helix pattern, there are some breakage will happen. So, that means in the strand like DNA, there is a strand S and there is a strand S prime. So, S and S prime will not be intact, they will be broken in the double helix pattern, and therefore, it is causing the damage to the DNA, and this once it is damaged, this DNA to RNA, RNA to protein synthesis cannot take place. Okay. So, this is known as the damaged DNA or fragmented DNA. Now, how these particles were generated? So, after sintering we get a sintered pellet. Now, this pellet can be crushed into small particles, then you can do ball milling, and in this ball milling process you can make fine particles. Now, these fine particles you can put it in the DMEM, that is the typical culture medium, you can disperse them, then you can simply filter them with a filter. After the filtration you get a particle size which is less than 0.22 micron. Okay, very fine particles, and this medium you can directly treat the different cells like L929 cells and other type of cells. So, this is the transmission electron image, transmission electron microscopy image, TEM image, bright field TEM image, and you can see this is 500 nanometer, means individual particles are somewhere between 70 to 80 or 80 to 90 nanometer. So, which is less than 100 nanometer. Here this is 500 nanometer, means individual particles here is greater than 100 nanometer. So, this is less than 100 nanometer, other one is greater than 100 nanometer. So, that we can see that what is the effect it can have as far as the toxicity is concerned. So, on the x axis it is the optical density is plotted, and along the y axis it is that different concentration of the hydroxyapatite mullite that is plotted here. Now, control means there is no particle treatment. So, in the control case all the cells are surviving. The moment you are adding the 10% to 100% concentration of the hydroxyapatite mullite particulates, then what will happen? You see after 24 or 48 hours the optical density decreases steadily. That means the cells are cells are becoming less and less survival. Okay. So, lower the optical density means lower number of cells are surviving in the culture medium. So, therefore, after long term, like after 2 days, that all these less than 100 nanometer particle size, that cells are dead, and then it shows some cytotoxicity. However, when the particle size is greater than 100 nanometer, what you see that you do not see that much drastic optical density reduction the way you have seen in the case of 100 nanometer, because here OD values is more than 80 and above 80. So, you cannot call it as a indication of the cytotoxicity. This is the case for the pure hydroxyapatite. As expected, you do not see any cytotoxicity. This is the case for pure mullite, and you normally see that is 80% or less, and above that 80% or 75% 100% mullite concentration. This is the typical cell morphology of the L929 fibroblast cells. What you see here, that cells are proliferated in some cases, but there is limitation of the number of cell proliferated when you are treating them with the mullite particles, and some small particles what we have seen, they are becoming internalized in the inside the cytoplasm of the cell. This is what you can see when you do some experiments, what is known as single cell gel electrophoresis. This is the nucleus of the cell, and on this nucleus, outside the nucleus, you can see the DNA is fragmented. If you remember that earlier one of the slides that I have shown you, that in the double helix pattern of the DNA, so this is the double helix pattern, right. Now, if that some part are broken, that means DNA is becoming fragmented, and once the DNA is fragmented, they also spread out both inside and outside the cells. Because why spreading out is possible? Because cell nucleus has also nuclear pore complex. That means there are certain pores are there, and through these pores this fragmented DNA can be outside the nucleus. Okay. Now, how to quantify DNA damage? Now, you have a certain tail here, you can measure this tail length, that is called DNA tail length, or you can measure the OTM. OTM stands for olive tail moment. Olive is the name of the scientist who defined that what is the tail moment olive has, this DNA this comet has, and then you can find out that what is the tail DNA length also. Now, if you measure this, you will see that this is the fluorescence image. This is A stands for control, B stands for 5%, 10%, 25, 50, 75, 100, and this is the positive control. This positive control means you can see extensive DNA damage here, lot of fragmentation. Negative control means no DNA damage. Okay. In between positive and negative, these two extremes, you see the moment you are adding 10%, 25%, 50%, 75%, that means that progressively more and more DNA being damaged here, and that is what is being shown here in this particular plot. You can quantify it, and then once you quantify it, you can see this is the less than 100 nanometer H20M particle size, and you can see there is statistically significant DNA damage, because there is a much higher tail DNA length compared to your control, which is negative control, but certainly this tail DNA is much less than which is known as the positive control. Positive control means this one. So, compared to this one, you do see that is a reduced value of the DNA fragmentation in these materials. This is the last thing that I want to discuss in the cell apoptosis. This is kind of very unique equipment which is known as fluorescence activated cell sorter or fax machine. What is the basic principle of this machine is that all the cells, they are tagged with certain fluorescence molecule. So, suppose it is a cell here. Okay. Now, there is certain fluorescence molecule here, and this fluorescence molecule are being attached to a particular cell here. Okay. Now, every molecule performs different function, I will come back to that. Now, this fluorescence molecule, the tagged fluorescence molecule, they comes with a cell stream. Now, at certain point they will experience certain electric field, and typical this electric field is that 2 kilovolt. So, there is a voltage difference, one side is positive, one side is negative. So, this is the 2 kilovolt voltage difference. So, depending on the molecules which are being tagged to the cell surface, certain cells will be separated from this time onwards this way, certain other cells will be separated from other test tube, and the cells which are unaffected by the electric field, they will go straight and then they will be accumulated at the bottom. Now, what are the type of cells they are being accumulated? This will be the towards the positive electrode. That depends on what kind of properties this fluorescence molecule has and how this fluorescence molecule are being attached to the individual cell. So, this uniqueness of this process, this is that one of the unique process which is also known as the which is also used in the cell sorting process. So, this is known as the cell collectors, these are like different cell collectors, and then before it is being before it is entering this electric field, there is a laser, this laser is focused on the cell stream, and once this laser is focused to the cell stream, this laser, depending on what is the population of the total cells which are still alive, that population can be detected. This is the typical results. Now, this annexin is one of the fluoromolecule, propydium iodide is another kind of fluoromolecule. Now, what is the uniqueness of this FACS analysis? This will tell in a given cell population what are the fractions of the cells which are alive. This is the L stands for alive. Now, what is the cells which are in the necrotic stage, that is N. What is the cells that are in the early apoptosis stage, that is E, and what is the stage and what is the how many cells are that is in the late apoptosis process. Let me summarize. You see here 4 quadrants 1, 2, 3, 4, okay, and the individual data points here, that individual data points are collected from one cell. So, each data point means one cell information. Okay. The second information, that second thing that you must remember is that these particular coordinates, the way it is mentioned, that is coming to this one. This particular coordinate stands for one of the collector tube. Okay. So, that how many cells that are being deflected in a particular directions, that individual coordinate data points is actually reflecting the cells in that particular collector tube. Okay. Now, if you go to the different coordinates here, if you go to different coordinates here, that means these data points means this many cells which are collected in a different collector tube. Similarly, these coordinates means this information being collected from some other collector tube, like that there are different collector tubes are there. Now, each coordinate has a particular meaning, and what is that meaning? This meaning is that in one of the coordinates, if it is the bottom one, then Pi is negative, propydium iodide is negative, and annexin also is negative. Now, what is this negative and positive they do in this FACS process? Okay, there are 2 cells that has been shown here. Left one is the vital or live cells, right one is the apoptotic cells. Now, in these 2 cells, this is called plasma membrane. This plasma membrane has a particular structure. As I mentioned while discussing the cell structure, plasma membrane has a typical double layer type of structure, means certain molecules are exposed to the extracellular space, certain molecules are exposed to the cytoplasmic space. So, this is your cytoplasm, this is your ECM. Okay. Now, what happens when a cell undergoes apoptosis? Some molecules which is known as that phosphatidylserine, that is the phosphatidylserine molecules PS, this is the red one here or different colored ones. These colored molecules, they are being increasingly more exposed towards the ECM compared to the cytoplasm. But when a cell is alive, you do not see any of the phosphatidylserine residue here in the outer leaflet, which is exposed to the extracellular matrix. Now, annexin 5 is a particular fluoromolecule. Now, what it does, this fluoromolecule has the specific binding capability to the phosphatidylserine. So, this when a cell is undergoing apoptosis, this annexin 5 comes, they are being attached to the PS molecules, and then more the number of data points you have, that means more number of annexin 5 molecules are being attached to the more number of cells during the cell apoptosis process. Okay. That means when the annexin 5 is negative, what it means? It means that the cells are live cells. When annexin 5 is positive and more concentration, that means more number of apoptotic cells are present. Is it clear? So, similarly, there are different there is another type of staining agent is that propydium iodide. What is propydium iodide? Propydium iodide directly intercalates with DNA and causing red fluorescence of the necrotic nucleus. Now, if propydium iodide is negative, what it means? It is live cells. If propydium iodide is positive, that means it is necrotic cells. So, similarly, if annexin 5 is positive, it is apoptotic cells. If annexin 5 is negative, it is simply apoptotic. Annexin 5 negative means live cells, annexin 5 positive means it is apoptotic cells. So, both the case annexin 5 positive and PI positive, that means cells are not anymore alive.